chapter. Now today we'll study about the megasporangium. Actually, detailed study we'll do about the pistil, which is the female reproductive part, as well as we'll study about the megasporogenesis, because it will give rise to the formation of the female gamete. Then we'll study about the pollination and the types of pollination, agents of pollination, and which one is advantageous and why, as well as we'll study about the merits and demerits of the two types of pollination and the characteristic features of the various agents of the pollination. That means the wind pollinated flowers, water pollinated flowers, and the insect pollinated flowers. As well as we will learn about the different outbreeding devices. So we begin with the chapter of your NCERT, page number 24. So now we'll first begin with the female reproductive part of the angiospermic plants, and we'll study here about the pistil, megasporangium, also termed as ovule, and the embryo sac. First of all, we must know now what is a pistil. As I discussed earlier in my previous lecture, pistil is also termed as carpel. It is a female reproductive part. Each pistil consists of ovary, style, and stigma. These are collectively termed as gynoecium. Now, the gynoecium may consist of a single pistil called monocarpellary, or maybe more than one pistil called multicarpellary. This you have learned in your class 11th also. Like a flower having a single pistil is called monocarpellary, and a flower having more than one pistil is called multicarpellary. Now, when more than one pistil is present, it can be in a fused state or it can be in a free state. This also you have learnt in your class 11th. Now, when the pistil is fused, it is termed as syncarpous state, and when the pistil is free, it is termed as apocarpous state. Now, further, we must know the presence of the syncarpous as well as the apocarpous ovule or the pistil. Here in the diagram you see different types of pistil. The first one, that is 2.7a figure, represents a dissected flower of hibiscus. Hibiscus, also termed as China rose or shoe flower. This pistil is of hibiscus. It is showing ovary, style, and stigma. They are placed on the base of the flower called thalamus. So you can see the another type of pistil in the B, 2.7b. It is showing you the multicarpellary pistil, but it is in syncarpous state of papaver. Syncarpous means, I am repeating, fused pistil. Then C diagram is showing you multicarpellary apocarpous pistil or the gynoecium found in Michella. Now again I am telling you, apocarpous means free. So the pistil is free, not fused. Then the D is the ovule, which I will discuss with you further in detail. Then again I will show you this diagram also. Now you have already learned the syncarpous pistil in your class 11th in your practical lab, where in the datura flower, where I had shown you the pistil is fused, and this condition of the pistil fused is termed as syncarpous and free is termed as apocarpous. Now we come to the different parts of the pistil. The stigma. It is the tip swollen where the pollen grains are dropped during the process of pollination. I repeat, stigma is a swollen tip where the pollen grains are dropped during the process of pollination. The style, it is long slender part, it is beneath the stigma. And the basal swollen part of the pistil is ovary. Ovary is very important part of the flower. Inside the ovary there is an ovarian cavity which is also called as locule or cavities. They bear a cushion-like structure which is made up of parenchymatous tissue called as placenta. I repeat, ovarian cavity bears a cushion-like parenchymatous structure called as placenta. Placenta bears one to many ovules called megasporangia. I repeat, placenta bear one to many ovules called megasporangia. Ovules in an ovary may be one, example wheat, paddy means rice, mango, etc. Or they can be many like in papaya, watermelon, orchids, etc. You can see ovules at your home also. Ovules after the fertilization gets converted into seed. So in any of the fruit or any of the vegetable, say for example tomato, in tomato you see the seeds arranged inside. These seeds were ovule before fertilization. After fertilization they have converted into seeds. So the ovule, now I show you the diagram, that is, please focus on the figure 2.7d. It is the structure of an ovule. This is an anatropous ovule. Meaning of the anatropous ovule is that ovule is completely inverted or turned back to 180 degrees on its stalk. I repeat, anatropous ovule is a completely inverted ovule turned back 180 degrees on its stalk. This ovule is large parenchymatous body. The parenchymatous body is termed as nucellus. I repeat, ovule is a large parenchymatous body called nucellus. It is formed in the ovary. The stalk of the ovule is called funiculus. The stalk of the ovary is called as funiculus and is attached to the ovary by placenta. The ovule fuses with funicle in the region called hilum. Ovule is surrounded by one or two protective layers. These are the outer layers called integuments, leaving a small opening at one end termed as micropyle. The basal part of the ovule where nucellus, integuments and funicle merge is called as chalaza. I repeat, the basal part of the ovule where nucellus, integuments and funicle merge is called as chalaza. C-H-A-L-A-Z-A, chalaza. The development of megaspore within the ovule is called as megasporogenesis. I repeat, the development of megaspore within the ovule is called as megasporogenesis. The ovule is composed of multi-layered cellular tissue called the nucellus. The cells of nucellus, they have abundant reserve food materials because they have to provide the nutrition to the developing eggs and the other parts. A hypodermal cell of the nucellus at the micropylar end differentiate and becomes transferred into megaspore mother cell. I repeat, a hypodermal cell of the nucellus at the micropylar end differentiate and becomes transferred into a megaspore mother cell. Thus, each diploid megaspore mother cell is formed from the nucellus. I repeat, each megaspore mother cell is formed from the nucellus by attaining the nutrition from it. Each diploid megaspore mother cell undergoes the process of meiosis. I repeat, megaspore mother cell is diploid, that means having 2n number of chromosomes. It will undergo the process of meiosis to form a tetrad of haploid megaspores. I repeat, megaspore mother cell undergoes meiosis to form a tetrad of haploid megaspores. That means the megaspores formed are haploid, that is having only n number of chromosomes. The formation of megaspores from the megaspore mother cell is called as megasporogenesis. I repeat, formation of megaspores from megaspore mother cell is called megasporogenesis. Out of these 4 megaspores, 3 megaspores degenerate. I repeat, out of these 4 megaspores, 3 megaspores degenerate and the remaining one becomes functional. And this functional megaspore enlarges at the chalazal end, which I told you earlier. I repeat, the remaining one megaspore which is functional enlarges at the chalazal end. The functional megaspores now undergoes 3 subsequent mitotic divisions. I repeat, the functional megaspore undergoes 3 subsequent mitotic divisions and forms 8 nuclei All the mitotic divisions are free nuclear type. All the mitotic divisions are free nuclear type and are not followed by cell wall formation Therefore, they do not develop the wall surrounding the cell. Now, the 8 nuclei arrange themselves into 3 groups. This figure you see of your NCERT, which is there on the page number, it is 26, figure is 2.8. It is showing you figure is showing you the parts of ovule, showing a large megaspore mother cell. This one, the first one, which is developed from the nucellar cell. The top one is a micropylar end, and you see the labeling nucellus made up of parenchymatous tissue provides nutrition to the megaspore mother cell. This megaspore mother cell, you see, it undergoes the process of meiotic division, I said. So in M1 it forms 2 cells, which is represented by megaspore dyad. I repeat, which is represented by megaspore dyad, which you see in this figure A. And the next figure you see here, it shows the megaspore tetrad. That means it is completed meiotic 2, giving rise to 4 megaspore tetrad, which I have just discussed along with you. So I am repeating, out of these 4 megaspore tetrad, 3 degenerate and 1 remains functional. Below you see, in the B figure you see the functional megaspore. I repeat, B figure shows you the functional megaspore, which undergoes the process of mitotic division. As I told you just now only, that it undergoes 3 subsequent mitotic division, which give rise to formation of first 2 cells, then 4, and then 8 cells. Okay, so 1, 2, 4 and 8 nucleate stages are formed. Now I would like to tell you that the 8 nuclei themselves arrange in 3 groups. These 3 nuclei migrate at the micropylar end and form the egg apparatus consisting of 2 synergids, which are the supporting cells, and a round egg cell centrally located. I repeat, 3 nuclei migrate at the micropyle end and form the egg apparatus consisting of 2 synergids, which are the supporting cell and a round egg cell. The other 3 nuclei arrange themselves at the chalazal end or at the chalaza and form the antipodal cell, which you see in the figure 2.8b last diagram, and the remaining 2 nuclei come together as polar nuclei in the central cell. I repeat, you see the figure carefully. The central cell is formed, which is containing 2 nuclei. These 2 nuclei come together as polar nuclei. They are also termed as secondary nucleus. This I had given some introduction in your class 11th also, but here I am telling you in a detailed form. So I repeat once again that the 3 arrange themselves as the chalazal end and 3 arrange themselves at the micropylar end, and in the central cell 2 nuclei come together as polar nuclei. The 3 cells at the chalazal end termed as antipodals, and the 3 cells at the micropylar end are termed as synergids and a round egg cell. Egg cell is a very important cell. Then you see some footprint like structures termed as filiform apparatus. These are formed from the base of the synergids. These help in guiding the pathway of the pollen tube inside this embryo sac. So I would like to tell you now further about the embryo sac. All the cells of the embryo sac are haploid. Please hear it carefully. All the cells of embryo sac are haploid except the polar nuclei, which is diploid, means synergids, antipodal cell, all are haploid, and as well as the egg are haploid and the polar nuclei is diploid. Thus the large central cell of the embryo sac contains 2 polar nuclei. A typical angiospermic embryo sac at maturity thus contains 8 nucleated and 7 celled structure. This is very important for the board point of view. The question also comes related with this, why the embryo sac is 7 celled and 8 nucleated. So I have made clear to you very well that it is 7 celled and 8 nucleated because in the central cell the 2 polar nuclei, they are not separated, they are fused with each other, they remain intact. Now during development of the female gametophyte in a majority of the plants, one of the megaspore is functional while the other 3 degenerate. I already discussed, and a functional megaspore develops into female gametophyte, that is embryo sac. This method of embryo sac formation from a single megaspore is called as monosporic development. I repeat, this method of embryo sac formation from a single megaspore, because 3 have degenerated, it is called as monosporic development. I will be providing you the notes of the same content, but the diagrams you can see from your this NCERT figure 2.8. Now this way the megasporogenesis is completed. Now we begin with the next topic of this content, that is pollination. Now what is pollination? This way you can come to know for the next topic, that is pollination. It is the transference of pollen grains from anther of a flower to the stigma of the another flower. I repeat, we have learned in your smaller classes also that pollination is the transference of pollen grains from anther to the stigma. That means anther of the flower to the stigma of the same flower, or it can be the transference can be to another flower of the same or different plants but of same species. So first of all, I would like to tell you about the types of pollination. The first one is autogamy. From the term itself you can come to know that autogamy means self. Auto means self, and it is also termed as self-pollination. Self-pollination, it is the transference of pollen grains from anther to stigma of the same flower or to the stigma of another flower born on the same plant. That means self-pollination can be on the same flower or between the two flowers of the same plant. When it is between the two flowers of the same plant, then it is termed as geitonogamy. I repeat, when the pollination occurs between two different flowers of the same plant, it is termed as geitonogamy. And when it occurs on the same flowers, it is termed as autogamy. Now we switch on to another topic, or you can say the another type of pollination, that is cross pollination. It is the transference of pollen grains from one flower to the stigma of another flower on a separate plant of species, or you can say it is also termed as allogamy. It is usually seen in dioecious plants. It is usually seen in dioecious plant which bring about genetic variations. In dioecious, I have already told you earlier also what is dioecious. So therefore here you can see the geitonogamy state. It is, I am repeating it, it is a condition where the pollen grains from one flower are deposited on the stigma of another flower of the same plant. Then this allogamy, it is a condition where pollens from one flower are deposited on the stigma of another flower born on different plants of the same species. Allogamy, it is also termed as cross pollination. It is also termed as cross pollination. Another term from allogamy, another term for allogamy is xenogamy. Here you can see in your NCERT, it is the transfer of pollen grains from anther to the stigma of a different plant but of the same species. Now, the transference of pollen grains are by means of certain agents. These agents can be abiotic or biotic. Abiotic agencies are water and wind, and biotic agents are insects, birds, bat, snails, etc. Now, what are the merits of the self-pollination, that is autogamy? Autogamy gives us greater chances of pollination, and it preserves the desired and parental characters of the plant. It is economical because large number of pollen grains are produced and it's like that it is going to take place because so many pollen grains are produced in large quantity, and it will, if 2 or 3 lose their viability, or some of them lose their viability, at least 1 or 2 are going to be transferred. So that is the benefit of the self-pollination. That means it is going to provide the parental characters, as well as there are more chances of autogamy in these flowers. The demerits are, it does not produce any new variety, means you are not going to see the variations, because here the parental characters are retained. It also loses the vigor. Vigor means the superiority and vitality of the plants and the seeds. I repeat, autogamy also loses the vigor, means superior features, as well as vitality of the plants and the seeds. And certain defective characteristics are not removed by the autogamy. I repeat, certain defective characters are not removed by the autogamy. In autogamy, the flowers may be open. Here you see above, it's written in your NCERT. There are two types of flowers. They are chasmogamous and clistogamous. I repeat, in autogamy, the flowers may be open. The open flowers are chasmogamous, or they remain closed and never open. They are clistogamous. You can easily learn, like clistogamous, they remain closed. So the spelling of the close is L. The L, that is of the clistogamous. So the spelling, the beginning two letters are similar, like close Cl and clistogamous Cl. So this way you can easily learn that clistogamous are the flowers which remain closed, they do not open at all, whereas chasmogamous, they remain open, they do not close, they are not closed. And this way the clistogamous flowers, they show assured seed set. Some plants produce both the types of flowers. Both the types of flowers are produced in certain flowers. Example, viola, camelina, oxalis, balsam. All cell pollination in a bisexual flowers occurs in bud condition only. I repeat, here the cell pollination occurs in the bisexual flowers in the bud condition, means when the flower is in bud state, bud, bud condition only, before the opening of the flower buds. Example, pea, wheat, rice, etc. Then I told you, we come on the agents of the pollination in detail. Here the plants use either the biotic agents or abiotic agents to achieve pollination. So pollination by wind is more common amongst the abiotic pollination. Now we come to the characteristic features of the wind pollinated flowers. The flowers which are wind pollinated are also termed as anemophilous, and the process is termed as anemophily. I repeat, wind pollinated flowers is termed as anemophily. Flowers, these flowers are odorless. Pollen grains are produced in large quantity as a result of lot of wastage. The pollen grains here are smooth and dry. They bear wings. Here the anthers are exerted, and generally unisexual flowers, they show anemophilous feature, and their stigma is feathery so that they are easily trapped by the airborne pollen grains. Example, maize, date palm, coconut, cannabis. Cannabis is also termed as bhang. Then the characteristic features of the water pollinated flowers. The process is termed as hydrophily. The flowers by water is termed as hydrophily. It is in completely submerged plant, that means in aquatic plants. Here the pollen grains are very thin, long, ribbon or thread-like, but they lack exine. The germinated pollen grains sink in water and affect the pollination of female flowers in the exile of leaves. Example, Vallisneria, Joystra and Ceratophyllum. In most of the water pollinated species, the pollen grains are protected from wetting by a mucilaginous covering. I repeat, in most of the water pollinated flowers, they are protected by a covering, the mucilaginous covering. Here you can see on the page number you see the page number 29, figure is 2.10. It is a wind pollinated flower showing compact inflorescence and well exposed stamens. This is telling you about the tassels. These are actually the tassels. So the wind pollinated flowers, they generally have single ovule in each ovary, and many flowers are packed into an inflorescence which is shown here. This is a figure of a corn cob, and the tassels you see, they are nothing but they are the stigma and the style. So this is important. Please underline that the tassels are nothing here but they are the stigma and the style. And since they wave in the wind, the pollen grains are easily trapped, and therefore this you can see in grasses also. Now we move to the Vallisneria. Vallisneria and Hydrilla I told you just now, that is Joystra, they are all aquatic plants. In Vallisneria, the female flower reach the surface of the water by a long stalk, and the male flowers or the pollen grains are released on the surface of water. Then with the water currents, female flowers, they reach to the stigma and the pollination takes place. In other sea grasses also the pollination is by water. That means it is hydrophily. And I already discussed the characteristic features of these pollen grains in already, means the characteristic features of the water pollinated flowers. They are, I am repeating, they are very long, ribbon like and the thread like. Then lastly we come to the characteristics of the insect pollinated flowers. The insect pollinated flowers, they are also termed as entomophily. Here the figure you see in the NCERT, this is about the Vallisneria, which I have already discussed with you. Now some other flowers, they get pollinated by different agents like bees, butterfly, that means they are insect pollinated. And the plants or the flowers which get insect pollinated is termed as entomophily. Here the flowers are very bright colored to attract the insects. They are very large and they are very showy. They are generally used as ornamental flowers. So I repeat, these flowers are very bright colored, very large and showy. Here the stigma and the pollen grains are sticky, so that when the insects get attracted to them, they come to consume the nectar and they get stuck to it. So this way the sign of the pollen grains, which is viscous and sticky substance, that is also termed as pollen kit, which I have already discussed with you. And these insect pollinated flowers, they show a good fragrance also, and just to attract the insects. Then many insect pollinated flowers, they can be of foul odor also, but actually the foul odor also, then also some of the insects, they get attracted to it, and they help in the process of pollination. Here you see the figure of the pollination of water in the Vallisneria above one, and the B one is the insect pollination. This is an orchid flower. Orchids are generally found in Himalayan areas or the northeast Himalaya. This orchid is having a special feature. In this orchid flower, one of the petal resembles a bumblebee. So the male bumblebee thinks that it is the female bumblebee, and this male bumblebee comes and sits on this petal. So this is showing you the pseudo copulation of the bee also, and in return this gets pollinated. I repeat, here the one of the petal of this orchid resembles the bumblebee. This bee is attracted by this, thinking that the female bee is there on the flower, and it comes and sits for the copulation. In return, the flower gets pollinated. It is also termed as, the flower is termed as ophrys. This you will study in your ecology also. So here you see that actually the insects are getting the nutrition, and it is a reward by the insects to the flowers, so that the flowers get pollinated. Now in some species the tallest flower is Amorphallus. The flower is about 6 feet in height. Here you can see even they can get pollinated, and you see the relationship between the species of the moth and the yucca plant. Actually they show the symbiotic association with each other. The moth lays egg inside the yucca plant or the yucca flower. In return, the flower get pollinated, and both of them, they show the symbiotic association, thereby helping each other. Now this further you can see in the figure of the yucca plant. So this yucca and moth, just on the top lines you see that yes, on the top lines you see the moth. I have already discussed you. The moth deposits its eggs in the locule of the ovary, and in turn it gets pollinated, and the larvae of the moth come out of the eggs as the seed start developing. So they show the symbiotic association with each other. Thank you, children.